Okay, hello again. They put us back in one of these wonderful rooms underneath the library. Okay, so just to start off, so Microbe of the Week, thanks everyone for sending answers in. Yeah, so here, this is the update on the scores currently. So the question was, can endospores survive in outer space, and how long, and how do we know? So plenty of people found out that in fact there have been experiments carried out on, I guess it's the International Space Station, right? So they took Bacillus pumilus or other Bacillus species endospores, put them outside of the spaceship for different amounts of time, and then pulled them back in and tested to see if they could still grow. And if they're protected from UV, they grow back pretty well, in fact. So they have reasonable survival after at least 1.5 years. And if you look a little bit further, there were also experiments done in the 1980s. They left the endospores outside of the spaceship for 6 years, and they could still grow afterwards. So it seems quite likely that endospores probably could, survive a trip from one planet to another. There's a couple of questions that so that raises another couple of questions, and this goes back to the old theory of panspermia, the idea that there are living things floating around in space and they can come and seed planets, and life starts on planets because of this. But that raises a couple of questions. One of them is, okay, so if UV radiation is so toxic, endospores on their own as a tiny little spore, they can't survive in space, right? So they'd have to be carried along by something bigger that's like a piece of rock, something like that, that's going to protect them from the UV. But then this rock is going to smash into a planet. So can they survive the impact? That's another question. So people have done experiments on that as well. And another point somebody made was that, yeah, so even if these endospores arrive on a sterile planet, they won't find good conditions for growth. So you need the reactivation and the germination stimulus, which will be absent. So perhaps it can't possibly work, or certainly we shouldn't really be too worried about, contaminating new planets with bacterial endospores. Or let's say you don't need to worry too much about that. So that was kind of the interesting answers that came from this one. And the second question was, on Earth, how old was the oldest endospore that has ever been reactivated? And there are really two answers to this question, because I kind of tried to look it up myself last week. So from about in 2000, there was a paper published where a group said they were able to reactivate endospores from a saltwater inclusion in a crystal of rock salt that was 250 million years old. Subsequently, after that, people have sequenced the genome of this thing because they were able to grow it up. And it doesn't look like it's a 250-year-old bacterial genome. It looks too close to current species of halophilic bacillus. Okay, so not everybody believes this thing was really 250 million years old. So, some people said 250 million years. Yeah, maybe not. And the previous record was people who got endospores out of a piece of material included in amber, and that was 25 to 30 million years old. People are pretty convinced that the bacteria that grew from that sample actually were conserved for 25 million years at least. So, under the right conditions, endospores can certainly survive for millions of years. Okay, so that's kind of the long introduction to today's stuff. Okay. So what we're doing today is to look at some of the very basic, very classical stuff that every microbiologist needs to really understand about the nutritional requirements of bacteria and how you use that to define different trophic types. What kinds of culture media are used to cultivate and grow bacteria, what the different kinds of media are useful for. What else do I need to say about that? Oops, I didn't want to do that, did I? Oh dear. It's okay. Yeah. And, oh, what am I doing here? Just totally out of control. Okay, yes. So let's get back into some semblance of knowing what I'm doing here. Okay, so what different types of culture media are useful for? And then we'll talk about, how to measure bacterial growth, how to measure bacterial growth rates, the different phases of the growth curve. And then that will probably be about halfway through the afternoon, around about here somewhere. And in the second half, talk a little bit about batch culture versus continuous culture. And something which is very interesting, which is kind of group behavior in bacteria. Normally you think, you've got these bacterial cells in a culture and each one is just dividing on its own. It kind of ignores that there are other cells around it. But that's not always true. So this kind of communal behavior in bacteria is very interesting to see how do they know about this stuff? How do they know there are other bacteria out there? So this is going to be quorum sensing and biofilms. And then the last part is going to be about these different environmental factors which can limit bacterial growth and how bacteria that can survive in, for example, conditions of high salinity, extremes of pH, extremes of temperature, and so on, can actually survive and resist these very, very difficult conditions. Okay, and I think I don't do them in this order. I think I changed this around. Okay, so nutritional requirements of bacteria. Well, it's just like for any other form of life. You need carbon, nitrogen, phosphorus, sulfur, and a bunch of metals, a bunch of mineral salts and oligoelements. So let's go through them one by one. So for carbon, okay, many bacteria require organic carbon. Could be carbohydrates, sugar, starch, cellulose, could be fatty acids, proteins, peptides. And in that case, all bacteria that require organic carbon are called heterotrophs. And there are other bacteria which can fix inorganic carbon, for example, carbon dioxide, like plants. And these bacteria are autotrophs. You get a similar kind of thing for nitrogen. So pretty much every type of bacterium can assimilate amino acids or purine or pyrimidine bases, so organic sources of nitrogen if they're available. And very many bacteria can also exploit inorganic forms of nitrogen. So going from the easiest chemical process to the most difficult chemical process, really huge number of bacteria. I'm not going to say all of them, but probably a significant majority of bacteria can incorporate ammonium, ions, or ammonia into amino acids just by a couple of simple chemical reactions. So this process is very, very easy, and almost all bacteria can mobilize reduced forms of nitrogen, ammonium ions, if they're available. Now, other types of bacteria are also quite frequent, not very, very rare, can use oxidized forms of nitrogen, nitrate and nitrite, and reduce these forms of nitrogen to ammonium, which is then assimilated into amino acids the same way as bacteria will use ammonium or ammonia. So once again, many bacteria can do this, but perhaps not so frequent as the bacteria that can mobilize ammonium, reduced forms of nitrogen. So when this process occurs, it's called assimilatory nitrate reduction, because the bacteria will have to put energy into the process to reduce nitrate, and that will be incorporated into the proteins of the bacteria. And lastly, some bacteria can reduce nitrogen gas, N2, into ammonium. So this is nitrogen fixation, and this is very rare. It's an extremely difficult chemical reaction to carry out. So, does anybody know the chemical process, the industrial process that's used to do this? With nitrogen gas reacts with hydrogen. You need to put it under a huge pressure, high temperature with a metal catalyst. Okay, it was called the Haber process, invented by Fritz Haber, very famous German chemist at the beginning of the 20th century. Probably a significant factor in Germany being able to fight on for so long during the First World War. Because, the British Navy had blockaded all the German ports in the North Sea. In order to grow enough food in the territory that they occupied, they needed fertilizer. Most of the fertilizer at that time, chemical fertilizer, was imported from South America. So this was blocked off. And so the only way they could grow enough food in Germany was through the Haber process, the Haber process. Okay, very important. Anyway, no, it's not the only thing he did in World War I. He was also behind the production of a lot of toxic gas used by the German army. Anyway, that's a little bit of a digression. It was just to say that, in fact, this is very, very difficult to get this reaction to work, because nitrogen gas is chemically extremely stable. And it's really some kind of amazing thing that cyanobacteria can do this, some species, and also rhizobium, which are symbiotic bacteria that grow in the root nodules of leguminous plants. And they can perform this extremely difficult chemical reaction at room temperature, just using the energy that they can extract from some glucose, or the energy they can extract from sunlight. So this is just an amazing thing that these bacteria can do. Okay, phosphorus and sulfur, also essential elements. So if bacteria are growing in an environment where you've got a lot of phospholipids, nucleotides, pieces of ribonucleic acid or DNA floating around, of course they can be assimilated and used by those bacteria. But pretty much most cells can also use inorganic phosphate. In all of our cells, we are producing ATP from ADP and inorganic phosphate. So direct use of inorganic phosphate is not difficult. Any bacteria can do this. Often it's a growth, it's a limiting nutrient for bacterial growth, phosphorus, which is why many bacteria can form polyphosphate inclusion bodies to stock up on phosphorus when there's a lot of it around. Okay, sulfur, it's present in 2 amino acids. So if you have a protein-rich medium, then bacteria will be able to get the sulfur requirement from those amino acids, methionine and cysteine. Otherwise, the incorporation of inorganic sulfur compounds is a bit like for inorganic nitrogen compounds. Many bacteria most bacteria easily incorporate sulfide into cysteine, just like one chemical reaction. Otherwise, many bacteria can also reduce sulfate or sulfite to H2S, to sulfide ions, and then incorporate the sulfide into amino acids, into cysteine. So this is assimilatory sulfate reduction. Now, in terms of other requirements, everybody needs, a fair amount of mineral salts to maintain, a constant osmotic environment inside the cell. So everybody needs sodium, potassium, and chloride. And also a bit of magnesium, because magnesium, as everybody knows, right, is essential as a cofactor for magnesium ATP. A lot of enzymes that use ATP, they use magnesium ATP. So, yeah, okay, also calcium is a enzyme cofactor for lots of other things, for other enzymes. As well as that, oligoelements, which are required in smaller quantities, smaller amounts than for, these like mineral salts where you need quite a lot. Oligoelements, these are metal ions, which are cofactors for enzymes that play some kind of essential role in bacterial metabolism. Now, the most important one of these is iron, because a lot of redox reactions in energy metabolism require, transfer of electrons with an iron molecule that's going to be 2+ or 3+. And that iron is either going to be in a heme group, a kind of porphyrin ring in cytochromes, or in iron sulfur clusters in other types of proteins. Once again, iron is often a limiting factor for bacterial growth, right? Because you need quite a reasonable amount of it to produce all these enzymes that are going to be involved in redox reactions. So you often need quite a bit of iron in a bacterial culture medium. The other oligoelements, you need much less, tiny trace amount, okay? Because they are cofactors for enzymes that, well, there's fewer of them, and the bacteria don't need to produce so much of these enzymes, okay? They're essential, but you don't need a lot of them. So you don't need a large quantity of these other oligoelements. And finally, some bacteria may not be able to grow just with these simple basic components. Some bacteria may require additional growth factors. So, for example, vitamins thiamine B1 and biotin B7, many heterotrophic bacteria which grow in a kind of like rich environment cannot produce, well, these vitamins or other components themselves. So what are these essential for? These are essential cofactors for different enzymes. So B1, thiamine, is used for a lot of biochemical conversions where 2 carbon atoms are transferred between 2 molecules. Biotin, this is essential for 1 carbon transfers. Now, yeah, so these may be essential factors for some types of bacteria. Other types, they can do without them, okay? So it's possible some bacteria can produce everything just from very, very simple components. But if they can't, then bacteria will be lacking the enzymes or the biosynthetic pathways that are required to synthesize certain vitamins or certain amino acids or purine or pyrimidine bases. And this is really the idea of prototrophs versus auxotrophs. So prototrophs for thiamine, for example, are bacteria that can produce thiamine themselves. You do not need to add it into the culture. Prototrophs for tryptophan synthesis. They can produce it themselves. You don't need to add it as a growth factor. auxotrophic bacteria, these are bacteria that cannot produce some particular complex molecule. So whenever you talk about bacteria that are auxotrophic, that means you have to say they're auxotrophic for something, okay? So if you can only grow bacteria in a medium that contains biotin, then the bacteria are auxotrophic for biotin. Now, I insist on this a little bit because bacteria that can use an inorganic source of carbon, they're called autotrophs. And that's a trophic type, and it's a totally different concept from auxotrophs. So you have two words that are almost identical, but they mean very different things. So you have to be careful, be clear in your mind what they mean and not to get them mixed up, okay? Okay, so that's basic growth factors and trophic types. So yeah, growth, yeah, nutritional requirements. So now let's go on to trophic type. So to define the trophic type of a bacterial species, bacterium, then it basically means you have to state how the bacteria gets its energy, its electrons, and its source of carbon. So if bacteria get their energy from light, they are phototrophs. So they are photosynthetic bacteria, right? If they get their energy from some kind of chemical reaction, they are chemotrophs. Electrons, well, the source of electrons can either be reduced carbon compounds, organic molecules, in which case bacteria are termed organotrophs, or some bacteria can extract electrons from mineral sources. You know, hydrogen gas, hydrogen sulfide, ammonium. And then finally, where do they get their carbon from? Either they require organic carbon, heterotrophs, or can can fix inorganic carbon. Now, carbon dioxide, bicarbonate in solution, it's the same thing. What kind of category would you put methanotrophs into here? Okay, methanotrophs are bacteria which can grow and survive with just methane, CH4, as their only carbon source. How would you put that? Would you put organic or autotrophs? I haven't prepared this as a question. How many people say heterotrophs? Nobody. How many people say they're autotrophs? Some people, most people don't care or don't know. Yeah, I kind of agree with the autotroph people, yeah?
Even if just autotroph means only for carbon dioxide.
Yeah, but this is the reason I'm asking the question. Okay, so so what does autotroph mean? Does it mean it's only carbon dioxide, or does it mean very...
One source.
Yeah. No. Very, very simple molecule. Okay, if you can grow just with glucose, okay, that's one source. You don't need anything else. But that's still heterotroph because it's a kind of complex organic molecule. So I don't know. Methanotrophs are a bit of a difficult one to put into these two categories because it's a very, very simple carbon molecule, CH4, right? And chemically, it's very difficult to make everything just out of methane. It's very tricky, but they can do it. On the other hand, it's organic. It's an organic molecule, so it kind of should be in the heterotroph category. So it's difficult. It's not easy to put them in. So people often use the term methanotrophs as a kind of like different category almost. Okay, because it's not really obvious where you should put them. But if you've got some bacteria that can grow just on like acetic acid or something, so this is a 2-carbon compound, you would call them heterotrophs. Okay. think that you only really have to worry about this for, methane. Yeah, okay, that's about all I've got to say for this. Okay, yeah, so you've got like 3 levels in the definition here, and for each level there are 2 categories. So overall, we should have like 8 categories, 2, 4, 8. Okay, so I've tried to illustrate that with this table. So first of all, I'm not sure the organization really works here, but what I'm trying to point out here is that, okay, here you've got organisms, bacteria that use a chemical reaction to get their energy. They use an organic source of electrons, an organic source of carbon. They are chemo-organo-heterotrophs. Bacteria that use light as their source of energy, an inorganic source of electrons and fixed carbon, they are photolithoautotrophs. Now, okay, so I'll just go through this. So, for example, cyanobacteria in green and purple sulfur bacteria are like this. There are also some photosynthetic bacteria that use light as a source of their energy, but they use an organic source of carbon, and the electrons that they assimilate also come from organic carbon. So these are photoorganoheterotrophs, green and purple non-sulfur bacteria. So in terms of autotrophs, we kind of understand what these guys are doing. They're like green plants, right? Okay, so they photosynthesize and they assimilate CO2. These ones are very interesting. These are chemolithoautotrophs. They're just living on rocks, basically. They don't even need light. Now, one of the things, okay, so photosynthetic bacteria, either using organic carbon or fixing carbon dioxide, bacteria that live on rocks, and then most of the bacteria that are in the world are heterotrophs, chemo-organoheterotrophs. So Escherichia coli, Staphylococcus aureus, Streptococcus, all this stuff that you grow in the lab, they're going to be heterotrophs. Now, one of the things that you might be asking, I know I was asking myself this, is, hey, I've got like 8 categories here, and I have bacteria that actually exist. They only fall into 4 categories out of the 8 possibilities. Now, why is that? Well, you have to try and think about what these categories mean. So, would it make any sense for bacterium to be a chemo-organo-autotroph? What does that mean? It means it's using some kind of chemical reaction. It's got organic carbon as a source of electrons, but it's going to fix carbon dioxide. That doesn't make any sense, right? So if you've got organic carbon hanging around already, there's no sense to use that organic carbon to get energy and then to use that energy to fix CO2. It's going to be inefficient. So this category doesn't exist in nature because it's just outcompeted by other forms of life, okay? And the same kind of argument works for the other empty categories. They just don't make sense, okay? See if I can do another one here. Yeah, it's the same with a photolithoheterotroph. So if you need to use photosynthesis, you use an inorganic source of electrons, but there's plenty of organic carbon around. So why don't you just get your energy from this organic carbon? So, okay, so that's why all these categories don't exist. You only have 4 categories. So that makes things a little bit simpler, okay? Right. Yeah, so just a couple of examples here. So, for example, Anabina, they're cyanobacteria, so they are photolithoautotrophs. Okay, the energy comes from light, the electrons come from water, and They're autotrophs, they fix CO2. Nitrosomonas, they are nitrogen oxidizing bacteria, so they use as a source of energy the oxidation of reduced forms of ammonium. So this is the electron source, and the energy comes from transferring these electrons onto oxygen. And they fix CO2, so they are chemolithoautotrophs. Yeah. And finally, most bacteria, like Escherichia coli, they use organic carbon as a source of energy, electrons, and, well, carbon. Okay, so let's see if... Oh, I'm pretty sure I had a question here. It's not there, it's gone. My question is gone. Ah, no. That's not good, is it? Ah, well, that's why I've got the wrong file open. Okay, so let's get this back. Ah, yeah, okay, so... okay, enseignement. Yeah. I opened the poly, which is the version without the questions in it. Yeah. Okay, so here you go. Just to make sure everybody is understanding this concept correctly. So I'll start the question up here. Okay, let's try and wait for 20 votes to come in. Okay, 25 votes. 28 votes. Okay, look, yeah, good. Okay, so here's what you voted for. And the winner is photolithoautotroph auxotroph for biotin. That's the correct answer. Okay. Photolithoheterotroph. This is one of those trophic types that doesn't actually exist, so it cannot be this. Photoorganoheterotroph, that exists, but there's no organic carbon in the medium, right? So that can't be right. Okay, you don't have any organic carbon here, so it can't be a heterotroph. It's got to be an autotroph. And okay, so you need light. You've got to be exposed to light, so it's photo. There's no organic carbon, so it must be getting the electrons from some inorganic source. It's got to be lithoautotroph. Then the last one, biotin. Does it need biotin? Yes, you have to supplement with biotin. So if your bacterium needs biotin, it's auxotroph for biotin. Okay, so that's why this one is correct. Now, do you think it's likely that a bacterium with these characteristics would live in nature? Who says yes? Yeah, we can go out in the, go out into the air and fish around a little bit and we'll get one like that. Nobody. Who says no? In no way, man. This kind of thing doesn't really exist. A few people. Okay. Right then, the few people who put your hands up, can you explain to everyone else why? Like turn around to the neighbors and tell them, all right? And all the people who didn't put their hands up, try and listen to what these guys are telling you, okay? Okay, not really into it. All right, all right. Right, because why am I saying this? Is because otherwise I'm going to tell you, right? But I don't think you will actually think about this any more than the first time I asked the question if I do that. Whereas if your neighbor sitting next to you is trying to explain this, then you might think about it a bit more. So please go ahead and talk amongst yourselves for 5 minutes. No, yeah.
I'm not sure, no, but I think that if we have the fact that it's a phototroph, it doesn't make a really big sense that it would be also a lithotroph. Because if I have a lithotroph, it should be in some I don't know, some stone sources.
Yeah, but litho just means inorganic. Doesn't necessarily. So it's going to be. So think about cyanobacteria. Okay, so it's something like that. So they're using, water as a source of electrons, not some kind of stone. Okay, so that's all it means. But photolithoautotroph, they exist.
Yeah, I'm sure that it is, but it's just the fact that I couldn't really imagine bacteria who could use also light and then be in some mineral sources. It's just
But the photo that is for the energy.
yeah.
Okay, but you have to get electrons somewhere because they're autotrophs, right? So they're using CO2 as a source of carbon to turn that into glucose, carbohydrate. It needs to be reduced. So you're going from CO2 to carbohydrate. You need to reduce it. In order to do that, you need electrons from somewhere. So that's why you need to have electrons. So the litho part is where the electrons come from, not necessarily the energy. Okay? So that's why it can be photolitho, because it's two
I
things.
don't know why it seems to be like this. I just, yeah. But okay, anyway, I will trust
Well, I don't know, maybe, yeah.
Is it because like biotin is like present in proteins like animals or red meat or something?
Yeah.
It cannot get light inside an animal. Like it's not gonna get light inside the meat.
Well, yeah, so somehow they're kind of incompatible. But if it's going to be living a life where you have a lot of organic, where are you going to find biotin? You're right,
Inside
somewhere
an
with
animal. Yeah, inside
some
something
animal or yeast
living.
or some other
Yes.
bacteria.
There's
So
like no light coming
if
inside.
yeah, but it might not be. It might just be in a kind of like lake or something with a lot of organic matter in there. So it might not. It might be available to light, but it still doesn't make sense. Okay, we'll see if we got some answers here. Okay, so everybody has had a little bit of a chat about this. So okay, so let's try and see what ideas you've come up with. So now, so the question is, a photolithoautotroph, which is auxotroph for biotin, is this likely to exist in nature? So, no, that's the question. Who says yes? Nobody. Who says no? Okay, most of you are convinced that this shouldn't exist. So, okay, why? Why doesn't it exist? So, could you give us your reasoning?
it is quite impossible to have all the factors reunited in the same place to have it.
Yeah, I kind of agree. I kind of agree with that. Anybody want to add a little bit of detail on that answer? Nobody else? Nobody got anything else to say? Okay. Okay. The reason this is unlikely, at least my point of view on this, is that if you are a kind of autotroph, photolithoautotroph, that means there's not much organic carbon around. This bacterium is photosynthesizing and it's producing organic carbon. There probably isn't very much organic carbon around. So if you've got no glucose, no fatty acids or something around, it's very, very unlikely that in that environment there will be some free biotin. So basically autotrophs, they tend to be able to produce everything themselves, all right? All the vitamins, all the amino acids, everything. Because they live in environments where they don't have organic carbon, okay? So when I was making this example up, I thought, oh no, this doesn't make sense because nothing like this could ever exist, in fact. Well, it's very unlikely to exist in a natural environment, but you could find bacteria like this in a research lab because the wild type, natural bacterium would be prototroph for biotin, but you could generate mutants where you've knocked out some of the genes for biotin synthesis. So this kind of phenotype could exist, but in a laboratory, okay? All right, so yeah, that's kind of what I wanted to say about the trophic types, prototroph or auxotroph. Right, so now we're going to think about different types of culture media. Starting off with defined media, also called sometimes a synthetic media or minimal media. In a defined culture medium, all the chemical components of the medium are precisely known and precisely defined. Okay. Oh, that shouldn't be shown here. So why is that? Oh, it's for some weird reason. Anyway, so this is a typical example of defined medium. So it's got glucose. It's got, okay, let's think. Have we got all of the mineral salts? Yes, we've got mineral salts here, sodium, potassium. We've got calcium, we've got with magnesium, we have chloride ions. Okay. We've got an inorganic source of nitrogen, we've got sulfur, we've got phosphorus. Okay, and we've got some iron in there. So if you put bacteria into this medium, if they are prototrophs for amino acid synthesis, vitamin synthesis, and purine pyrimidine base synthesis, they can grow in here, okay? So it works, but if you think about it, it really shouldn't work. So why shouldn't it actually work, this type of medium? And then afterwards we try and figure out why it does work. Oh, come on, stop. Gotta start the question up. It's not open yet. Okay. For people at the back, I thought I'd like get the screen and pick it up like this to make this move up a bit higher. And I thought, oh no, that's not going to work, is it? Okay, we've got 25, 26 votes coming in here. I have to say, I can hardly believe what I'm seeing here. Yeah, let's do that again. Okay, right, so let's have a look and see what you think. All right, so basically, this is the clear loser here. Only 2 people chose this one. And we got a bunch of people choosing these other 3 things. So since there's a kind of split opinion, well, let's do this talking thing again, okay? So I would like you to find somebody who made a different choice, gave a different answer, and try and explain to them, oh yeah, I chose this, I think it's right because whatever. And then the other person will explain to you why they chose something different. And then after you discuss with each other, maybe you can realize one of you was wrong, or maybe both of you were wrong, you know. Okay, so do that for a while, and then I'll ask everyone to re-vote and see how it looks, okay? Let's try that. So you've got to tell everyone what you voted. Yeah, find your neighbours, find someone who voted something different. Oh, go back to 0. Yes. Ah, sorry. Say, yeah.
Sugars.
Okay, you guys in the back, not much talking going on around here. Normally, the back of the class is where you go if you want to talk, right? so, or you finished, you decided everything. No? Yeah, well, discuss with them. Maybe they have a different idea. Oh, okay, I got it. Thanks. Okay, so if you've like had a bit of a discussion, everybody can revote, and we'll see what it looks like. See if we've got a consensus. Okay, 19. Okay, I wonder if I could like. What I need to do here is, yeah, I need to stop the presentation. I need to put this on over there. All right, just keep the question up. Okay, let's have a look at the answers. So I'm a little bit surprised here. I think there are a lot of people in the class here who should definitely go into politics because you're very persuasive, but very good at persuading people to accept the wrong answer. So, okay, what are the answers that come through? We've still got number one, say this medium doesn't have any organic carbon. Glucose, there's your organic carbon right there. Oh, yeah, come on, they're not like total diabetics, these bacteria. Yeah, 1 gram a litre, it's not much, but you can grow, right? You can grow. Oh, okay, so it's... No, that's enough. It's enough to grow bacteria, okay? Okay, so answer one is wrong, okay? There is some organic carbon in this medium. So what else is wrong here? It lacks a source of organic nitrogen. That is actually a true statement, where this one is just downright, well, it's just false, right? This is just downright false. This is actually a true statement, but it shouldn't matter because you've got ammonium salts here, right? And almost all bacteria can assimilate ammonium into ammonium salts right here, right? Almost all bacteria can assimilate ammonium into amino acids. So this is true, but it doesn't matter. Okay? It's not really a restriction on growth. So answer 3, it lacks growth factors. Well, that's also a true statement, okay? But what I said is that when I was talking about this, is that prototrophs, bacteria that can synthesize all of their own amino acids, vitamins, everything, they can grow in this kind of medium. So in fact, it doesn't matter. Okay, let's say it doesn't matter, but it does. Only certain bacteria can grow in this, okay? They have to be prototrophs for everything. But I said that when I was talking about this. So 3 is not correct. That leaves number 4. This is the right answer. In a couple of slides before, in the oligoelements, you know, I had weird stuff like manganese, molybdenum, cobalt, this kind of stuff. These metal ions are essential oligoelements, but they are not anywhere in the formulation of the medium. So how come it still works? How can bacteria still grow here? Any ideas? Okay, I'll give you a slight clue here. If you look, I should actually find this, but if you look in the catalogue, when you want to make up some of this medium, and you look at the supplier's catalogue, you'll find, you know, calcium chloride. You'll have different grades of calcium chloride, which have hugely different price per kilogram. And generally speaking, the cheaper it is, the less. Anybody know the word I'm looking for here? The less pure it is. So if you use a relatively cheap source of calcium chloride, magnesium sulfate, ammonium sulfate, they'll be maybe 99% pure or 99.5% pure. And you'll have a whole bunch of other contaminants in there, including some trace amounts of the oligoelements. So in fact, that's why bacteria can still grow in this kind of medium, even if you haven't put any manganese in there or any molybdenum, because it's all present, it's already present as a trace contaminant in all of these inorganic salts. Okay, and that's not entirely obvious. I think it's, you know, but you have to think about it in order to reach this conclusion. Okay, so if you will, so what would you have to do if you wanted to show that some bacteria require molybdenum? What would you have to do? Any idea? Okay, another way to ask this question: what kind of chemical compound would you have to order? The very cheap one or the really expensive one? Yes, the expensive one, because you would need to make the medium with constituents that are as pure as you can possibly get them. So you make sure you don't have any tiny trace amounts of oligoelements. And then you could add back in some different metal ions and find out what bacteria really need. Okay, so that's basically the kind of utility of this kind of medium, because it allows you to study what the nutritional requirements actually are of the particular bacterium you're looking for. So imagine you've got a bacterium that grows pretty well on LB medium, which we'll just see in the next slide, and you try and get it to grow in a minimal medium like this. It doesn't work. Then you can say, okay, I'll add in some vitamins. Still doesn't work. Okay, I'll add in some vitamins and then some amino acids. Okay, it works. And then you can actually define what the nutritional requirements are for that bacterium. The minimal medium supplemented with different stuff. Okay, that's enough about that. So what else? Okay, so next one to think about is a complex medium. And an example of a complex medium is LB. So it's got peptones. So these are proteins from meat, casein or soya that have been hydrolyzed. So it's like a lot of peptides, yeast extract and salt. It's called a complex medium, not because it's difficult to prepare. It's very, very easy to prepare. But it's complex because within these components, you have a complex mixture of a whole bunch of stuff, and you don't exactly know what is in here. But basically, within the yeast extract, you'll have, okay, within the peptones, you'll have all the amino acids required for proteins, and within the yeast extract, you'll have pretty much every vitamin the bacterium would like to have, okay? And so with this kind of complex medium, you can culture a lot of chemoheterotrophs that don't have any particular nutritional requirement. So you would call them non-fastidious. These bacteria are not fussy. Now, next thing is an enriched medium, which is a complex medium, and you've added something that contains some extra nutrient that the bacteria you are trying to culture really need. And one which is quite frequently used in medical microbiology is fresh blood agar. So you'll have some tryptones, peptones, so hydrolysate of proteins, salt, agar, and sheep's blood. And that's very useful to culture things like Listeria or Streptococci, which don't grow very well on LB medium, for example. I have a picture here. Yeah, there you go. So there's a Streptococcus pneumoniae colonies on, okay, Streptococci. Yes, many Streptococci will grow well on LB medium. Streptococcus pneumoniae does not grow very well. So Streptococcus pneumoniae colonies here on a fresh blood agar plate. So you add something, these are bacteria that are going to be pathogens. They might need some kind of molecule that's only present in, there's a lot of it in eukaryotic cells, so you put some blood in there, and it helps them. Might be something to do with their requirements for iron, in fact. Okay, selective media. This is almost like the opposite of enriched media. It's a complex medium, but you've got some components in there that are going to prevent certain bacteria from growing. And you can also have elements or components that favor the growth of the bacteria you want. So you are selecting against the bacteria you don't want and selecting for the bacteria you want to grow. And you'll be using this anytime when you're inoculating from a complex source. A patient's diarrhea or sputum or something like this. There are going to be a lot of bacteria in there. And the ones that are interesting might only be 1% or less of this whole mix. So if you use a selective medium, you can get rid of a lot of the background. Okay, so let's take a look at some selective media. So Chapman medium is what you'll be using in the lab classes. The selective component is a high concentration of salt, so only halophilic or halotolerant bacteria will be able to grow. So for example, Staphylococci, they can grow in here, but Enterobacteria will generally, most gram-negatives will not be able to grow at this high salt concentration. Now another selective medium for Staphylococci is Baird-Parker medium. And this contains lithium chloride and tellurite, which are inorganic compounds which are toxic for most bacteria. So they inhibit the growth of most gram-positives and gram-negatives, whereas Staphylococci can actually resist these, can grow in the presence of these inorganic compounds. There's a second selective component, which is the presence of pyruvate and glycine, which are used selectively by Staphylococci as preferential sources of carbon. So most other bacteria will prefer glucose. So you have two components here. The tellurite and the lithium chloride will inhibit most other bacteria, and the organic carbon source will be helping Staphylococci to grow. And finally, so these are both selecting for Staphylococci for gram-positives. McConkey medium is a medium used for selectively allowing gram-negatives to grow because it contains bile salts and crystal violet, which will inhibit the growth of gram-positive bacteria. Now, many selective media are also differential, and that means that the medium contains some kind of component components that allow you to differentiate between the different types of bacteria that grow on the plate, on the agar plate. So Chapman is also differential because it contains mannitol. So, you have to know this, but in fact, most strains of Staphylococcus aureus are mannitol positive. So if you get, oh, I didn't know this. No, I'm going to have to take that away afterwards. Okay, so, yeah, I'll put it up and take it away. So here's a plate of Chapman agar. You have these bacteria here. They don't ferment mannitol. They don't acidify the agar, so the agar plate remains red. Here, this is Staphylococcus aureus, ferments the mannitol, the agar becomes acid, and the indicator turns yellow. So that's giving you information. And okay, Baird-Parker. There are two differential agents in here. So the tellurite itself is interesting because when it's reduced to detoxify it, it becomes black. So black colonies indicate the reduction of tellurite. And halo, JLo, oh dear. So the other differential component is the presence of egg yolk in the medium. And the lecithin in the egg yolk can be digested by lecithinase activity. And this combination of characteristics, tellurite reduction and lecithinase activity, is very specific for Staphylococcus aureus. So Staphylococcus aureus colonies on Baird-Parker medium look like this. Okay, a black colony with a clear halo. And Baird-Parker is the reference technique for quantifying the presence of Staphylococcus aureus in the food industry. And finally, for McConkey agar, the differential component is the presence of lactose, and this will allow you to differentiate between bacteria that can ferment lactose and those that can't. So gram-negatives that ferment lactose are coliforms. So you'll have bacteria like this that can't ferment the lactose. They give clear colonies, cream-colored, and bacteria that ferment the lactose give pink color. Okay, so differential and selective media, very, very often used in routine microbiology whenever you want to isolate and directly identify certain types of bacteria from a particular source. So you have to know what you're looking for, right? There's no point in using Baird-Parker medium to isolate Escherichia coli. Won't work, okay? These selective differential media only work for certain types of bacteria. And you choose the medium you want to use depending on what bacterium you are looking for. Okay, I have another question coming up now, so okay, have a think about that one. Wait. Okay, 10 minutes. Oh, really? Oh, the problem with the it only accepts one answer. Oh, okay, I'm sorry. Fine. Okay, well, let's see what people wrote. Is that right? It's only you can only choose one option? Ah, okay, sorry. I better fix this now because otherwise I'm going to forget. But okay, I'll fix it afterwards. So let's see. We wrote, okay, we'll do these. So that's what you were able to send it. So defined or complex, it's complex, right? Because you've got peptones, meat extract, complex mixtures, you don't know everything that's in there. And then for this selective and differential thing, okay, so it's either differential for gram negatives or salmonella, or selective for salmonella and gram negatives. So pretty much everybody figured this one out. Yeah, this is the correct answer. It's differential for salmonella because you can have salmonella and shigella that grow on there, right? So only the salmonella colonies will be black. So it's differential for salmonella. And it's not selective for salmonella because it's not only salmonella that can grow on here, okay? It's selective for gram negatives because it's got, what have we got? Bile salts, okay? Bile salts is the selective agent here, which just like in the McConkey medium, will inhibit the growth of gram positives. So it's selective for gram negatives, differential for salmonella. Okay. Right, we have about 5 minutes, and I think we better use them, and then, oh, I don't know. Actually, I think it'd be a good time to break now, and then we'll come back and talk about bacterial growth curves and then the environmental factors. Okay, so can everybody try and be back in 10 minutes at 25, not 3:30? Thanks.